This talk is brought to you by the Bamberg Mathematical Consciousness Science Initiative.
Oh, thank you so much for having me. It's a real pleasure and privilege to share some ideas with all of you. Yeah, as you know, I don't primarily work on consciousness, but I thought I would say a couple things as well, and then we can discuss. If anybody's interested in digging more into some of the things I'm going to say, because some of them you may not have heard before, and if you want to see the primary data that underlies these claims, everything is here at our website. All the software, the data sets, the public, peer-reviewed papers, everything is here. And this is my own personal blog around what I think some of these things mean. So what I want to talk about today is really this notion of diverse intelligence and the issue that a lot of the problems that are coming up now, very interesting problems in this field, are not really new with AI at all. They're, I think, just reflections of very deep fundamental questions about ourselves that remain unsolved, and the AI is just making them stark at this point, and all of that is discussed here. The interesting thing is that we as humans, and this is the sort of thing that figures prominently in chapters and papers and so on, and books on philosophy of mind, and they say, well, the human, and what they typically mean is a modern adult human like this. But we are at this intersection of two continua. One is both on an evolutionary scale and a developmental scale, that we all evolve from cells, from single cells. And whatever you think this human has, you actually have to have a story about how it got here, and where, if anywhere, does it stop. And then there's another continuum, which is through biological and technological changes, where again challenges this to say, well, what do we actually mean when we say human? What's actually important about this? Is it the DNA? Is it the standard organs? Is it, what is it that's actually important? And this is the sort of thing we're going to be dealing with, not just language models sitting in a box somewhere, but human cyborgs and hybrids and different kinds of beings that we're going to have to be able to interact with. So what I do not claim for today is to have a new theory of consciousness, nor do I have any definitive data uniquely supporting one of the existing theories of consciousness. But here's what I will say, plus some other stuff, that use of cognitive tools and frameworks well outside the brain has led to some really useful discoveries, new capabilities. It has empirical fertility, which tells us that it's probably a good idea to start using those tools to look at things that are unconventional. The other thing that I will say is that if you use any of the typical criteria by which we give each other the benefit of the doubt that we are conscious, of course, you don't feel my consciousness, but we give each other the benefit of the doubt for a few typical reasons that people use. If you like any of those reasons, you should take very seriously the idea that there are other body structures also have consciousness. And if that's the case, then of course you might think further, and where you would like to cut it off and why. I think that AI and many problems in ethics and so on are not going to be resolved if we maintain our focus on humans, on three-dimensional space for embodiments, on brains, and on crisp binary categories, which I'm going to try to argue against. And in the and towards the end, I'm gonna talk a little bit about maybe the most controversial part of what I have to say, which is that I think neither physics actually nor computer science as we know it, nor evolutionary theory as we know it, tell the whole story. I think these are actually good theories of interfaces, and I'm going to try to say something about what I think they're interfaces to. So the vast majority of the talk, I think I'm on fairly solid ground with years of data, and then towards the end I'll speculate on some things. So here are the kind of basic pieces of today's talk. And what I want to do is highlight some tools that we have for trying to overcome what I think of as mind blindness. So prior to a good theory of electromagnetism, we thought there were all kinds of disparate phenomena. So we had lightning, and we had magnets, and we had light and static electricity. We had all this stuff, and we thought these were all different things. These were categories, fundamentally different things. And then we had a theory that not only conceptually unified those as aspects of one underlying phenomenon, but also enabled us to operate in a range of, in fact, a spectrum of those phenomena to which we were completely blind because of our own evolutionary history. This is all we can see. We didn't know about any of this other stuff until we had proper theory and the technology to exploit it. And I think exactly the same thing is the case with the spectrum of minds. I think we have a lot of mind blindness because of our own history. So the first claim that I'll make is, if you take modern evolutionary theory and developmental biology seriously, we have to say that the continuity thesis is the null hypothesis. In other words, there are no sharp, drastic sort of categorical changes as you move forward here. Certainly, you don't see anything like that in developmental biology. If somebody wants to make a case for emergent phase transitions, things that are binary, such that, well, over here at the beginning, you didn't have something, whether it's consciousness, intelligence, whatever it is, and then boom, now you have it, and there's a sharp phase transition. Maybe, but somebody's going to have to make that claim with strong evidence for what the phase transition actually is. The background should be the continuity thesis. In other words, a story of scaling. We should be trying to understand what happened to get from here to here. What's the parameter that scales, and how does it scale, and why does it scale? Oftentimes people, I emphasize this because oftentimes people see this backwards. They think that it's obvious that this is in a different category, and that we really have to argue for this continuity thesis. And I think the basic facts of biology here are telling us that it's the other way around. And of course, then there's this spectrum, which again, it makes it clear that these kind of stories that people tell, well, life does this, and machines do this, and computers are never going to do X, Y, Z. All of those kinds of things also require very strong specification of what exactly the difference is. And it's made even harder by the fact that biology is very interoperable. And so pretty much at every level of organization, you can make these hybrids where you mix evolved and design materials as much as you want. And all of these terms, which come to us from past ages, sort of pre-scientific times, they're not remotely as helpful as they used to be. And I actually don't think they're going to survive the next few decades. And so we have to understand what actually is changing here, and how. So my framework that I'm working on, the goal of it is to enable us to recognize, create, and relate to truly diverse intelligences, regardless of what they're made of or how they got here. This should include familiar creatures. Whoops, what just happened? Sorry. Oh yeah, okay, here we go. Oh, that's weird. Familiar creatures such as primates and birds and things like that, but also unusual things like colonial organisms and swarms, and then engineered synthetic life forms, AIs, whether purely software or robotic, maybe someday aliens, and then some really exotic things that we can talk about, such as patterns within media. Two things to note about this. This is not primarily a story of consciousness, as I said. However, if I really don't think we can have any kind of a proper theory of consciousness without understanding intelligence, which is sort of the third person accessible kinds of problem solving competencies. If we can't really get our minds around that in novel substrates, I really don't think we're going to be able in any position to say anything deep about consciousness. So I like to start here. And the other thing about this kind of program is that it's very important to me that this is not a philosophical project or a linguistic project to try to redefine terms. This needs to be empirical science. It has to move experimental work forward, has to give us new capabilities, better ethical frameworks as well. So in other words, all the stories I'm going to tell you today, the only value that any of these models have is if they actually make research move forward. That's my sort of strong criterion. And I'm not the first person to try for something like this. So here's Rosenbluth, Wiener, and Bigelow in 1943, trying for some kind of, this is of course a model of some sort of great transitions, but the idea is how you get from passive matter all the way up to human level metacognition and so on. And my version of this in detail is here. So one of the things that I think we're going to need to do in this field to make progress is to really start to expand our perspective. And I want to talk about, give you some examples of what I mean by this. And for this, I'm just going to illustrate this model system. So we got this caterpillar. So this is kind of a soft-bodied creature. It has a controller suitable for driving a soft body. It has a very particular kind of brain. It lives in the two-dimensional world, crawling around, eating leaves. It has to become this kind of creature, which is a butterfly, hard body, completely different controller needed for that, flies around in a 3D world, and the brain is very different. In order to get from here to here, what happens is that the brain is basically dissolved. Most of the cells are killed off, all the connections are broken, and it gets rebuilt, and there it is. So one of the remarkable findings, and you can see many examples of it here, is that if you train the caterpillar, so let's say you train it to see a certain color stimulus and then crawl over and eat some leaves, what you will find is that the moth or butterfly eventually forms still has those memories. Okay, behaviorally, you can tell that it still remembers. Now, one amazing thing is, okay, how do you store memories when the memory medium is being refactored like this? That's amazing in itself. But there's a deeper issue here. The issue is that the actual memories of this caterpillar are of no use to the butterfly. It doesn't crawl, doesn't care about leaves, it likes nectar. Everything is different, it has different visual system, everything is different. And so you can't just maximize the fidelity of the memory, you have to remap it to keep saliency to a completely novel embodiment. So here are three perspectives on this system that would be interesting to take up as we think about AI and what's going to happen and all of that. First is the perspective of the caterpillar. The caterpillar is facing a singularity. It is going to cease to exist as a caterpillar, but through certain modifications, it's going to have some kind of a different life in a higher dimensional space. It's going to care about new things that it doesn't care about now. It's going to have new capabilities. How should it feel about that? If it could, that's one perspective. Another perspective is that of the butterfly. And so the butterfly is born, and it has this beautiful embodiment that it can use and do all these things, but it's saddled with some behavioral programs that it inherited. In other words, it has these vague memories, in fact, functionally important memories of needing to do certain things when it sees certain stimuli. Where did those come from? It has no idea. It inherits them. It doesn't know where they come from. It's not from this life that it's having. It's some other process in its history that saddled it with these things. So that's the other perspective. And then there's a third one, which is the weirdest one of all, and I think it's important, and that's from the perspective of the memory itself. Now, we are all patterns in excitable media. We are metabolic patterns and some other kinds of patterns, and you can imagine that memory itself existing in the excitable medium of the brain and body of the caterpillar. So now that memory is facing the paradox of change. If it wants to persist, it has to change, and it has to make itself useful to the nervous system and the embodiment of the butterfly, and and maybe even do a little niche construction and sort of modify the brain a little bit to make it easier for it to be held onto and things like this, right? So there are numerous perspectives here that we can try to take on to gain some mental flexibility, and that's important because this is actually the journey that each of us took. We started off as a little blob of chemistry and physics, so this little unfertilized oocyte. Through a slow and gradual process of morphogenesis, we became something like this, or even something like this, that's going to say we're not a machine. And then there's some other things that can happen actually even after that. But it's important that we took this tour through all the sciences. So here we were, the province of physics and chemistry, and then eventually, sort of physiology and developmental biology, and then behavior science, and then eventually psychoanalysis and psychiatry. So we toured through all of those, and there was no magic point where you stopped being physics and you started being a cognitive being. This is a slow, gradual process. So whatever is happening here, we have to understand the story of transformation. Not is it or isn't it, but what kind and how much. And it's critical to know that we are all collective intelligences, not just the beehives and the ant colonies, but all of us. This is the kind of thing we're made of. Now, this is a free living organism called the Lacrimaria, but you get the idea. It's a single cell, no brain, no nervous system, lots of competencies at its own single cell level agenda, and we are made of this kind of thing, right? We are like all intelligences, we are made of parts, and like biological intelligences, our parts are themselves quite competent. Now, you look at something like this, and I might say, again, from the perspective of consciousness, could we reward or punish it? Could we do things that it either really likes or really doesn't like? And I think most of us would say yes. It's pretty clear that we could do things that it would not be neutral towards. It would have valence around those experiences. In fact, this right here is somebody filmed the story of the death of a cell, and you can see it. You can see it here. It's basically one of these creatures, and then eventually it falls apart. And there, right before your eyes, something is happening. Again, you could zoom in and make this as slow as you want. This is not a rapid, it's not a sharp process. You could drag this out for as long as you want, but something happened here where this is no longer the same kind of creature as this was. And if you don't think that, if you think that you can reward and punish this kind of thing, and if you feel bad watching this die, which I certainly did the first time I saw it, then I would ask you the second question, which is, if we just had a chemical pathway, just a set of chemicals turning each other on and on, like a gene regulatory network or a molecular pathway, could you reward and punish a chemical network? Most people I ask say no. And that's weird, because guess what those cells I just showed you are made of? They're made of exactly this stuff. And in fact, what we found is that you don't need a cell, you don't need a nucleus, you don't need neurons, even simple molecular networks, okay, so small networks of genes describable by coupled ordinary differential equations are capable of several different kinds of learning, habituation, sensitization, even Pavlovian conditioning. And we're taking advantage of all that in the lab to do drug conditioning and other things for biomedicine. So we are some kind of collective intelligence made of components that are made of cells, which are themselves pretty competent. They are made of a chemical, of a material, a chemical substrate, which itself has competencies, right? So all the way down. And in fact, something interesting we found recently, which is that when you do train these molecular networks, their causal emergence goes up. And in fact, there's this amazing positive feedback loop. It's like an asymmetric ratchet that the more it's starting actually before you have reproduction selection. This is, I think, at the very base of evolution, that for free, from the laws of mathematics, and we'll get back to this at the end of the talk, you get this amazing ratchet where the more you learn, the more you are reified as actually an integrated agent, and higher your integration, the better you are at learning. So there's this like closed loop that points upwards in terms of agency all the way from the bottom. So this is the kind of embodiment that we have. Now let's for the final thing for this section, let's look at intelligence and embodiment. I think it's very important to remember that as humans, we are obsessed with three-dimensional space. Our sense organs are, sort of pointed outwards into that space, and we find it relatively straightforward for medium-sized objects moving at medium speeds, like these crows that are picking up cigarette butts to get rewards, for us to recognize that this is some kind of an intelligent process. Okay, we're okay with that. But at different scales, and especially in different problem spaces, we start to lose that ability. And it's important to remember that biology has been solving problems by navigating spaces long before we had nerve and muscle. So even single cells navigate the space of possible gene expression, the space of physiological states, and then multicellular collectives navigate anatomical morphospace, which we'll talk about. So all of these spaces have agents in them that are executing the loop that's really important about embodiment, which is sensing, processing, decision making, and then finally actuation. That loop is done in all of these other spaces. It doesn't have to be rolling around in three-dimensional space. So when we talk about AIs being disembodied, we have to be really careful, whether it's AIs or whether it's brain organoids or something sitting in a dish. Everybody says, well, that's not a real mind. It's not embodied. Yeah, that doesn't mean all that much, because there may be other problem spaces which it's traversing and navigating using exactly the same kinds of cognitive processes that we use to navigate three-dimensional space. Okay, so what I want to talk about next for a few minutes is a model system that we have where we try to understand a kind of semi-alien collective intelligence. And it's important because I don't think we're well equipped yet to study really alien minds. because we have enough problems with things that are almost like us. And I'm going to show you an example of this. And already this is considered, pretty wild and completely unconventional in developmental biology, even though this is a set of agents that basically share evolutionary history with us. The only reason they're alien is because they're operating in a different space, and they're operating in a space of anatomical outcomes, and that's enough to throw people off and to have a really hard time thinking that this really is a kind of intelligence. I should say that actually some people got it, and Turing was, I think, one of the first. He doesn't need any introduction to this audience, but interestingly enough, so he also wrote this seminal paper called The Chemical Basis of Morphogenesis, where he tried to understand how embryonic order could arise from chemical disorder. And it's very interesting to think about why somebody that was interested in intelligence and novel embodiments for mind and reprogrammability and things like that, why was he interested in chemicals in early embryonic development? And I think it's because he saw a very profound truth, which is the deep symmetry between the way our bodies and the way our minds come together. And so this is something I've been digging into for years now. And I used to have my students do the following: take a neuroscience paper and simply do a find replace, in Microsoft Word. Every time you see neuron, just say cell. Every time you see milliseconds, make that minutes. And all these terms about behavior in 3D space, just make it about morphospace. And then you get a perfectly reasonable developmental biology paper. And we now have a tool. You can go here and play with it. We now have a tool that does this for you automatically, because there are massive symmetries between cognitive neuroscience and developmental biology. And again, in fact, evolution is telling us that the symmetry between what brains and bodies do, that is the null hypothesis. The mechanisms are all conserved. If you want to consider these as different subjects, different disciplines, and many people do, because most of my colleagues in developmental biology and genetics and so on do not want to say that their material is problem solving or doing any kind of cognitive processes. They would like to describe it with the laws of chemistry, but I think that's a fundamental mistake, and would actually need a lot of justification. The null hypothesis is the symmetry, and it's really worked out well for us. So I want to point out what's actually happening in the creation of our bodies and of our minds. When we look at an early embryonic blastoderm, so let's say there's, 100,000 cells here during this stage, we look at that and we say, there's an embryo. Well, what are we counting when we say it's an embryo, one embryo? What is there one of? There are hundreds of thousands of cells. What is there one of? Well, what there's one of is alignment to a specific region of anatomical space that all of the cells are committed to going. There's a collective that is consistently aligning its parts. Okay, so there's a level of organization that is aligning its parts by giving them signals that basically tells a very compelling story, aka model, that is going to drive them to reach a particular outcome in anatomical space. And one of the things you can do, for example, is, and I used to do this in duck embryos as a grad student, you can take a little needle and put some scratches into this blastoderm, and until they heal up for the next 4 to 6 hours, each of these islands can't feel the presence of the others. It thinks it's the only one, and so it self-organizes into an embryo, and when you're done, you have 3, okay, or 4, or however many. So now a couple of interesting things here. First of all, we see now the origin, basically at boot up of an agent. What's happening is that the pieces have to be aligned. And in fact, how many agents are going to come out of this is not obvious. It's not determined by the genetics. The way you get twins and triplets and quadruplets and so on is because this material, this excitable medium here, can fragment into numerous different different large-scale agents. And so how many beings can come out of an early embryonic blastoderm is not fixed. It could be anywhere from 0 to half a dozen or more. And of course, each one has to figure out, where do I end and the outside world begins, right? So what are the boundaries between them? And all of this has fascinating parallels in cognitive science, because again, if you want to ask how many human minds fit inside a human brain, the answer is not obviously one, because dealing with split brain patients and dissociative identity disorders and all of those kinds of things, we find out that actually there's potentially dozens of human-like agents in there, if not more. And the question is, you know, what is the mapping between the actual medium and the number of beings that are there. In computers, we kind of know how to add up, at least add up the amount of bits you can store in a piece of material. But here the situation is much more interesting. And I want to show you. I don't have time to talk about all of the actual data, because but normally I would give a whole separate talk on how all this works, because we've made considerable progress in it. And I just want to show you a couple of quick things. So this is going to be a video of cells. All of these things are cells. The colors are voltage, that each of these cells have voltage, not just your neurons. Every cell in your body has voltage potential, and they form electrical networks that comprise the cognitive glue that binds cells together into a larger scale system that navigates this anatomical space. Just like you and I are collective intelligences using bioelectricity of our neurons bound together into a network that navigates three-dimensional space, these electrical networks navigate anatomical space. And this frame, this is before and after. So you can see here that this cell has a different voltage right up until this guy touches it right here. And that's all that it takes to convince the cell to join the collective. So here's the video. So here this guy comes crawling along, minding his own business. This one is going to reach out, boom, that's it. All it takes is that tiny little reach out. The voltage changes to green, and now it's part of this collective, and now it's going to do the thing that it already does. A very convincing message, bang, right there. Now, we would like to understand what that message was. And again, we have some progress in this, because we would like to give different messages. So for example, for regenerative medicine, which is mostly where all of these things play out in my lab, is we test these things by using them to develop new therapeutic candidates for birth defects, regenerative kind of approaches, cancer, and so on. I'll just show you one example of many. This is a tadpole right here. So it's a side view. Tail is back here. Here's the gut. Here's the right eye. The left eye is back here in the plane of the screen. And then here's the brain and the mouth. So what we've done is inject some ion channels that pass a particular voltage state onto these cells. And it so happens that this voltage state says to the cells, make an eye. Okay, and when you do this, sure enough, that's what they do. Gut cells make an eye, and you can do this anywhere in the body. You can convince them, if you put in the right message, you can prompt them to make an eye. I call it a prompt because really it relies on the intelligence of the material to do this. We don't tell it how to build an eye. We don't talk to the gene expression. We don't talk to the stem cells. We don't tell it how the dozens of different cell types and tens of thousands of genes that have to be controlled to do this. We don't do any of that. We give a very high level prompt to a system that understands messages about organ level events, not molecular biology, not even cell level biology, but organ level events. And we say, build an eye, and it does. These eyes will have all the right internal components, as you expect. The material is actually really interesting. It has all kinds of other competencies. For example, this is a cross section of an eye being formed somewhere. Look, the blue cells are the ones we injected. Okay, but there's not enough of them to make an eye. So what do? They actually convince all these other guys out here to participate in the process. So much like other collective intelligences like ants and termites, they have the ability to recruit others to the task that they've become committed to. So this is the kind of thing that we would like to do: communicate to the agential material of the body, not try to force it, not micromanage it, but communicate goal states to it, and hopefully make very complex things happen that we don't know how to micromanage. Now, one of the interesting things about that material is the following. And again, this is I'm highlighting a symmetry between what happens in cognitive systems and what happens in the material of our bodies. Consider the following example. Here's an axolotl. So it's a little amphibian. If you take a tail and graft it, and this is from a paper in the 50s, if you graft a tail to the side of this animal, what will happen is that tail over time is going to remodel into a limb. Okay. Now take the perspective of the cell sitting at the top of the tail. Something remarkable is happening. The local environment is perfectly fine. There's nothing wrong here. There's no injury. There's no damage. Why am I turning into fingers? Okay, I'm a tail tip cell sitting at the tip of the tail. Everything seems fine. Why am I turning into fingers? Well, because at the local level, at the molecular level, and also at the level of cells, there's nothing wrong. There's no error. There's nothing to do, and everybody's happy. But there is a higher level that dominates, where the cellular collective at the level of the organism knows perfectly well that you don't have tails in the middle, you have limbs. And so what it does is send signals that transduce all the way down the hierarchy and actually make the molecular biology adapt to a very high level kind of abstract goal. No individual cell knows what a finger is or how many fingers it's supposed to have or anything like that, but the collective absolutely does. And that's what it's doing, is making the chemistry and physics dance to that tune. Now, this is something that happens in your body on a daily basis. Imagine you wake up in the morning and you have social goals, financial goals, research goals, whatever it is. In order for you to get up out of bed and execute on these incredibly abstract kinds of goal states in your mind, ions have to move across your muscle cell membranes. The chemistry has to be altered in order for you to execute on these goals. So the normal cycle of voluntary motion, and this isn't some crazy yoga mind matter kind of thing. It's the everyday miracle of voluntary motion when extremely abstract goal states in a mental space that no cells understand, and certainly the ions in your membranes don't understand anything about it, but your body transduces these incredibly abstract goal states into making sure the chemistry actually works the way it's supposed to do to make it happen. And so in our lab, we're trying to communicate with all these different transduction layers because it's mostly bioelectricity. But the idea is that groups of cells solving morphogenetic problems, such as being deviated here from the correct pattern, is really a powerful model system for understanding collective intelligence. And this is something that happens in all of us. Something else that's another symmetry that's important for us is the following. Just consider first let's look at the cognitive version, and we'll look at the developmental biology version. The cognitive version is this: at any given moment, okay, so right now, at any given moment, you don't have access to the past. You have no way to access the past. What you have access to are memory engrams. These are the encoded, compressed results of past experiences, cached out in some sort of molecular, bioelectric, whatever memory structure, and that's it. You actually don't have access to what really happened. You have these memory traces that at every moment you are now responsible for interpreting. Now, you have to interpret it because the process of compressing past experience throws away lots of data, right? So inevitably you get this bottleneck. So you guys all recognize this autoencoder looking thing where you have a bunch of individual experiences. There's a process which squeezes it all down to a specific memory that is kind of generalized across everything that happened. And this side is algorithmic. So there are algorithms for squeezing these things down. But this side inevitably is creative because you've lost information. And in order to figure out what this thing means, you have to decide, and this is again and again, every 300 milliseconds for us or so, you have to reformulate a story of what your memories mean and what is your model of yourself, what's your model of the outside world. Now, under normal circumstances, that doesn't change too much minute to minute, but it's important to know that memories are a constructive process. Your sense of self is constructive. This is, I'm not saying anything new here. This is just, modern neuroscience. This idea that there are no non-destructive reads, that every time you access a memory, you're actually changing it because it is this process of active interpretation. So we are, in a sense, a self-telling, self-constructing story that propagates through time. And your interpretation of these things does not need to be exactly the same as past versions of you interpreted it. In other words, memories are messages from a past self. Like any message, they have to be interpreted. Maybe you interpret it the same way as the sender meant it. Maybe you don't, because your only allegiance is to the future. You want to tell the most adaptive story that you can, not the same one that was there before. So it's not the fidelity of the information, it's the saliency of the story and the adaptive value of the story. And this is what we really are, is like this continuous kind of a process of continuous improvisation, or confabulation, depending on how you want to call it. And this is extremely important in biology because, as I'm going to show you momentarily, biology does this too. So we all start from a little egg. Okay, the same bottleneck. We don't come, bodies don't build other bodies. Bodies build eggs, which are this incredibly compressed representation of the experience of past instances of the lineage. And then embryogenesis is not just a hardware, kind of a hardwired process by which the DNA encodes things. That is really not the right way to think about this. And you can see that that in this paper, where we really explain that the process of mapping this initial prompt, so you can think of the genome as a set of prompts, to a process that is fundamentally a creative, intelligent, problem-solving process that under normal circumstances will give you the thing you expect. So typically dogs have puppies and cats have kittens because the process has been evolutionarily shaped such that under default circumstances, it always does the same thing. But that isn't all that it knows how to do. And when circumstances change, it is perfectly happy to solve the problem in completely different ways. Okay, and so this idea of being this like self, where the intelligence is and maybe the consciousness too. You know, maybe consciousness is what it feels like to be in charge of interpreting your own memories. You know, when there's no other user, no one's there to tell you what your memories mean. Maybe that's an important part of consciousness. Is there at the basis of life, and it's there in our cognitive system too. This is essential, because if this process wasn't creative and wasn't a problem-solving process, maybe there used to be life that didn't do that, but it isn't around anymore, because you know, biological material is very unreliable. This is a very unreliable medium, and the only way that any of this is going to work is if this process is not trying to hardwire anything that's happening here. Evolution doesn't make specific solutions to specific problems. It makes problem-solving creative agents. And I'm going to show you, okay, and so next, for the next few minutes, what I want to show you are some examples of synthetic morphology, meaning we're going to take some perfectly standard cells, and I'm going to show you what the material is capable of under novel circumstances. If the goals that it was trying to meet, meaning making specific bodies, if those goals cannot be met, what does it do? And what it does is find new goals to pursue. And this is important. These things are going to be important for our attempts to understand consciousness. That's and the reason that we like the synthetic morphology business is that otherwise we fall into this trap of, if you ask why does this creature have specific an anatomical property, specific behavioral properties, you say, well, evolution, selection, that's what it was selected for. So of course it has those. So we want to go to a place where that argument isn't going to work, because I think it takes us to a much more interesting place. So I first introduce you to something called xenobots. The way these come about is we take a perfectly normal frog embryo. Okay, we don't touch the genome. There are no synthetic biology circuits here, no scaffolds. 8 hours later, it makes some epithelial cells up here, which are going to form skin and some other stuff. And we cut those off and we dissociate them here. Now, they could have done many things. They could have died. They could have crawled away from each other. They could have formed a flat layer like cell culture. Instead, they make this interesting kind of thing. And I'll show you first how that happens if we zoom into this process. Okay, so these cells coming together here, each one of these circles is a single cell, and this is a little blob of, I don't know, probably 15 cells or so. Okay, and I just think it's awesome that it's shaped like a little horse, but not all of them are shaped that way. So here it is. It moves as a collective. It's over here like trying to attach itself to this thing, and there's a little bit of interaction here. Then you're going to see a little calcium flash, which is just a sign of cellular computations. And this is what these guys actually look like. They repurpose. They have cilia, which are these little hairs that wave on the outside of their bodies, typically used to move mucus down the body of a frog. But in this case, they use them to row against the water. So here it is swimming along. They can go in circles. They can patrol back and forth like this. They can have collective behaviors like that. We can make them into weird shapes. So you punch out the middle, you can have a donut. And I'm just going to show you two interesting things that they do out of many. One interesting thing is we call it kinematic replication. So if we take these xenobots and we provide them with lots of little loose materials, so these other epithelial cells here, they basically do von Neumann's dream, which is a machine that makes copies of itself from stuff it finds in the environment. Because what they do is they run around and they collect the cells into little balls, and then they polish the little balls, both collectively and individually. They polish the little balls into these kind of things. And then the little spheres mature into the next generation of xenobots. And guess what they do? They run around, do exactly the same thing, and they make the next generation and the next generation. So what we have here is a weird kind of replication where we've made it impossible for these things to reproduce in the normal froggy manner, but that's okay. They found a different way to make copies of themselves. Something else that's interesting is when we asked about the transcriptome, what genes do xenobots express that are different from what they would have been expressing inside the embryo, we found hundreds of genes that are expressed differently. And one of the clusters had to do with sound perception. We thought that's weird. Why are they expressing genes related to sound perception? Is it possible that they be responsive to sound? And so we put a speaker under the dish. We played sounds to them, and we found out that yeah, in fact, as you provide audio stimuli, they do change their behavior. For frog embryos don't do this. This is only xenobots do this. This is a new kind of thing, and we studied this whole question of the transcriptome of an entirely novel organism. Okay, what genes does it express, and why are they expressing, all of a sudden they're expressing audio genes? Now you might think that this is something special to frog cells or embryo cells. This seems really weird that they're able to do this. So I would ask you the following question: what do you think your cells would do if we liberated them from the virtual governor of the rest of the body? So I introduce you to another model called anthrobots. So this little thing looks like we got it from a pond somewhere. It looks like a primitive organism. Here it is close up, again with cilia on the outer surface. If you were to sequence this, you would find 100% Homo sapiens. These guys come from adult human tracheal epithelial cells. Again, no genetic editing here, no synthetic biology circuits, no scaffolds or anything like that. This is what your tracheal cells would do if we took them out of your body and gave them a chance to reboot their multicellularity. So what do they do? Well, we can see they're sort of autonomously swimming around, and they have a little, they have certain kinds of movement patterns. If we place them on a plate of human neurons with a big scratch through them, what they do is they collect into a kind of a superbot cluster here, and they start to knit together the sides. They're able to heal the neural wounds. If you lift them up, this is what you see. Now, who would have thought that your tracheal epithelial cells, which sit there quietly in your airway for decades, dealing with mucus and whatever, that liberated from the rest of your body, they would come together into a self-motile little creature that looks nothing like any stage of early human development, and that would have some, and that would be able to go around and heal neural wounds, right? What else do they do? Well, a couple of other interesting features. They have 4 different types of behaviors, and you can build a little transition diagram of probabilities. Not 12, not 1, exactly 4 behaviors. They have 9,000 differently expressed genes than they did when they were sitting in your body. So about half the genome is now radically changed in its expression. They're younger than the cells they come from. So that's pretty cool. And we have, of course, an anti-aging project in the lab trying to understand what happened to them. How did they get younger? Because in this process of assembling into an anthrobot, they reverse their age, which is kind of neat. And so here's why I'm showing you all this. It's really interesting to ask, where do these come from? In other words, here's the genome, frog or human or whatever. We know when it learned to be a tadpole and a frog, right? The eons that it was bashing against the environment to select for specific features. When did it learn to be a good xenobot or a good anthrobot? I just showed you some extremely specific things, 4 behaviors, healing neural wounds, 9,000 specific genes that are differently expressed. When did it learn to do all this? When was the computational cost paid for this? Because there haven't ever been any xenobots. There's never been any anthrobots. There's never been selection for any of this stuff. Where does it come from? And you could, of course, say that, well, it's sort of emergent. Well, what does that mean? Well, it kind of happened at the same time that you selected for a frog, you also somehow got this. And in a certain sense, that's true. But the problem with that is that it undermines the whole point of evolutionary theory. The whole point is you should be able to tie the features of a creature with the environment of selection forces that it had faced over time. That's the whole point of evolution, is that specificity. And so now we see that, yeah, you can sort of select for this, but in a different circumstance, you can get something completely different that's never existed before. And we need to understand that something is missing here, because no one saw this coming, and no one thought that we could make anthrobots, and certainly no one predicted those specific features. So we need to figure out where they come from. So now here comes, so everything that I've said up until now, I think is pretty pretty much on solid ground. And so what I'm going to do now is make some very kind of speculative claims about how I think we should think about these things. And here's the thing. When we ask, so when I show you creatures that have never been here before, right? You can't just say it was selected for that. So it's not selection. We do need to understand why those specific features and the space, option space from which these features were chosen. And typically biologists like two kinds of things. They like genetics, meaning a past history of selection, and they like physics. In other words, certain things are just determined by physics. You're round because you're trying to minimize, surface area, whatever you're trying to do. So where else, if it's not going to be, if it's not going to be physics and genetics, because these things have never been around before, where else do specific patterns come from? Well, there's one other source, which is math. And this particular image here is a Halley plot of this very simple formula in complex numbers. Okay, if you plot this is what you get. All of this rich structure, and it also doesn't hurt that it's very organic looking, but all of this rich structure, and in fact we can make videos of these by slowly changing the parameters, and we can kind of do a flyover of that space. All of this comes from this little formula. But notice something important. There is no history here. This wasn't selected to be this way from years of evolutionary optimization, nor is there any aspect of physics that determines this. In fact, as far as I can tell, you could change anything you want in the physical universe. You could change all the constants at the Big Bang. You're never going to change this. This is what it is, and this information is not. It's not derivable from nor changeable by laws of physics. It is its own sort of source of properties of various mathematical objects. So of course, again, here, I'm not the first person to say this. Heisenberg and Whitehead and many other people had going all the way back to Pythagoras, and probably long before him, suggested this idea that there is a non-physical space of patterns, so the kinds of things that mathematicians study, and that these patterns affect what happens in the physical world. And what the claim that I'm going to make is, I think we call physics those things that are constrained by these mathematical patterns, okay? And biology, I think, are the things that are what we call things that are actually elevated or potentiated by these patterns. In other words, biology exploits the heck out of these things. I think that as far as evolution is concerned, I think that all these patterns in this space of mathematical truths are a kind of free lunch, where you can get some really interesting capabilities without actually having to evolve any of the details, because you're handed the patterns and you get out of it more than you put in. There's something else interesting here, which is that for both in physics and biology, if you ask why long enough, you just keep following an explanation, then why this, eventually you always seem to end up in the math department, right? And I'll just give you a very simple answer. So you're a biologist, you want to know why these guys come out at 13 and 17 years, right? These are cicadas, they come out at 13 and 17 years. Well, the answer is it's because they're trying to not to coincide with predators, right? Because if it was 12 years, then every 2 year, every 4 year, every 3 year, 6 year predator would get you. So say, okay, that's pretty good. So, but why 13 and 17? Oh, because they're prime. Well, why is that? Okay, now you're out of biology, you're out of physics. Now you're in the math department trying to understand the distribution of primes. And this happens, as far as I can tell, all the time. If you keep pulling that string, eventually you're down to some property of numbers or topology or the amplituhedron or something. You're always back there. So here's how I like to think about this. Where do the properties of novel creatures come from? And I promise I'm going to tie all this back to the consciousness issue and the cognition and consciousness issue. Where do these properties come from? Well, the first option, and this is what I think most scientists like, is they want a sparse ontology. They don't want to think about the Platonic spaces or any of that. They say, look, the physical world is all there is. And these weird things that you're finding, and the fact that gene regulatory networks can do Pavlovian conditioning, and all, and the fact that they rise their causal emergence, these are just facts that hold. We'll call it emergence, basically to indicate that we were surprised when it happened. We didn't see that happening. So we didn't see it coming. So we're going to call it emergence. And there's just a lot of facts that hold in our world, and that's it. The good news is that then you have a sparse ontology. You can keep being a physicalist. The bad news, I think, is that it's a very Mysterian view. It just basically means that every time we get surprised, we're going to call it emergence. If we eventually understand it and it becomes obvious, then we say it's not emergence, and we'll just catalog these things, and when they show up, they show up. I like a very different way of thinking about it, which is basically what the mathematicians do, which is to assume that it's not a random grab bag of features that you find in these novel creatures. There is an ordered latent space of possibilities, and it is structured, and we can systematically investigate that space by using these kinds of things that we build, which is, of course, embryos, but also biobots and all kinds of synthetic constructs. We can use them as vehicles to navigate that space. That's the research agenda, is to map out that space. Okay, and so again, this is something that's been said by many people, that mathematical truths are not just random stuff that we either find or make up, but rather we are discovering an ordered, structured space of these things. And so up until now, this is all, again, I think not particularly new or weird. But now here comes the really strange thing. I'd like to extend this in the following way. If we've already said that there is a space of mathematical truths which informs, literally informs, it constrains physics, and biology exploits these things, and so on, we can say that, okay, math is the kind of behavioral science of one specific layer of that space, meaning simple things that obey the kinds of formal models that mathematicians like. But what else is in that space? Are we sure that that's the only thing that's in that space? And could it be that there are other patterns within that space that have much more complex, dynamic, context-sensitive behaviors that actually behavioral sciences would recognize as kinds of minds, right? And we've even had, so we're doing this symposium on the Platonic space that's running all fall, and we've had a couple of mathematicians who even sort of chided me for claiming that the mathematical objects themselves are low agency, as that, they said that, well, that's just as much a prejudice too. You should do experiments and find out what they're capable of, and I completely agree. It's these things. This is not a philosophical project. This is an empirical set of questions to ask. What are these patterns are actually capable of, and how many of them look like behavioral propensities? In other words, things that really are kinds of cognitive architectures. And so the model is basically this, that mathematics studies one layer of this space of patterns, these kind of simple, well-behaved, relatively simple, well-behaved things. But there are other patterns here that are the kinds of minds that we see in cognitive science. Now, at this point, you're probably saying, okay, this sounds like we're back to Descartes, which was a real problem. This idea that you have some sort of non-physical patterns, that's some kind of a contents of a mental world, which somehow affects the biology of the brain and thus behavior. Isn't this a fundamental problem? Because what the problem was, and this is, of course, Descartes was criticized for this heavily. It's this issue of, how does the interaction happen? How are you going to have a non-physical pattern interact and make actual functional changes or causal changes in the brain? I would just point out that physicalism, I think, was already dead even in Newton's classical world. Never mind any quantum mechanics stuff, but even in the classical world, it was already functionally affected by patterns that were not themselves physical. In other words, I think it's been known since the time of Pythagoras that the physical world is not closed, and I would hypothesize that epiphenomenalism is as bad in cognitive science as it is in mathematics, because these forms actually matter. They're functional. And I would say that the mind-body relationship is basically a scaled up version of the relationship between mathematical truths and physical objects. Okay, the same kind of ingression that you're getting here. If you weren't freaked out about the interaction here between mathematical truths and the physical events that have to obey them, then basically this is more of the same, just sort of on steroids. And there's some interesting data that need to be thought about on this, which is this notion that if it is the case that certain behavioral propensities are actually coming from another space, we can start to ask this question. And of course, there are lots of ancient kinds of systems of thought that propose this idea that the brain is basically a receiver. You know, it's a thin client. It's not the actual show. And there are, of course, modern neuroscience doesn't like this idea at all, but we still have to deal with some clinical cases where there are humans walking around that have very little brain volume. In fact, they're missing most of the structures that neuroscience tells us are important for various cognitive capabilities. And in fact, they have normal or above normal IQ. And Karina Kaufman and I reviewed all of this here. You can look at them here. It's really, really astounding examples. So lots more research needed here in this kind of thing. So the final, and then I'll stop. I'm already over time. So I'll just say one final thing, which is that, okay, and this is typically where I lose anybody who's been hanging on so far. This is typically where I lose them. Some people are okay with this idea that, all right, biology is very complex. It has some sort of secret sauce that we don't understand. And what it's doing is serving as an interface to these amazing patterns from Platonic space. And yes, biology is great at it. And so that's why we're special. That's why biology is unique. And I just want to point out that I really don't think that's it. I think whatever is going on here with where these patterns are ingressing into various kinds of embodiments, I don't think it takes life or huge complexity or anything like that. I think it seeps into absolutely everything. And we can talk more about that during the Q&A. But this is one, just very briefly, I point you to one example, and we have more on this coming out in a couple of months on even simpler systems, where we took a look at something that everybody thought they understood. Every CS student studies these sorting algorithms, right? For whatever, 60 years, everybody's been studying these things. And we looked at them in a slightly different way without changing the algorithm whatsoever. And we noticed that some very interesting things were happening that are basically not in the algorithm in any obvious sense. Yeah, they certainly sort the numbers like they're supposed to, but they also do these weird side quests, and we can get into that were neither prescribed but nor forbidden by the algorithm. They do things in the spaces between what they were forced to do, and I think that's very important for us. And so this is what I'd say. That a lot of people are comfortable saying that living things, especially humans, are the full story of life and of humans is not to be told by the rules of biochemistry. So somehow there's something more going on. Many people are comfortable with that for humans, but at least it's okay. There's this area of the world, these machines, which only follow the algorithm. They only do what you force them to do, and at least they're well behaved. Biology is doing weird stuff, but at least the algorithms tell the whole story of what the machines are doing. And my point is, no, the formal models never tell the story, the entire story of the whole thing. You know, I think this is what Magritte was telling us. This is not a pipe. Well, this is not a Turing machine. This is a formal model of one. And I think that minds are not fully defined by our models, neither the limitations nor the competencies. And I suspect that everything we know about algorithms, about the thermodynamics of computation, about biology, are the theories of the front-end interface. In other words, I think the things we make are interfaces for front-end clients for patterns from the space, which is where a lot of the action is. So the implications, and then that's it. I'm wrapping up. The implications. I'm not sure there are any machines in the way that we thought there were, or dead matter, or anything like that. I think, for example, when we do when we try to train these things, and we're basically doing a kind of Skinnerian conditioning, which is very, very limited. I think we force these things to do certain things, like language for LLMs and so on. But if even a dumb bubble sort has these intrinsic motivations that nobody saw coming, more complex systems certainly will have them too. And what they're doing may have nothing to do with what the actual algorithm is. So I have a feeling that when we make these things, we're fishing in a brand new area of the space that maybe has never been embodied before. And we can say some interesting things about freedom that's completely different from either predictability or randomness, and so on. And so I think the reason this is important is that pretty much every combination of evolved material, engineered material, and software is going to be some kind of viable agent, some kind of a cyborg or hybrid or some kind of chimera that's going to be hosting a bunch of patterns, many of which we've never seen before. Like this is, everything we know on Earth, all the life on Earth is like this tiny little corner of this enormous space. And when you make novel embodiments, you will meet new beings, and we have to get better at understanding what's going on to be able to form some kind of an ethical synthbiosis with these things. So that's it. Just to summarize, here's what I feel pretty confident of, is that in this field, we should not be using binary categories. We need models of scaling and transformation. I don't think it has anything to do with brains or even embodiment in 3D space. I think that biology is telling us how rich the input from this latent space is, and our formal models, we need to remember that they're just formal models. They do not tell the whole story, neither for life nor for supposed machines. And then there are some things that we're actually working on. We're trying to understand what aspect of the interface is actually important for figuring out for determining what you get. We're trying to quantify how much free lunch there is. There's some wild work that's coming soon about how much you can actually get more or less for free in this way. I don't know if evolution is really important for this. Richard Watson thinks it definitely is. I'm not sure. I think that synthetics can take advantage of this just as easily. And then there are many things that would be fun to talk about, but I really have no idea, around what all of these things mean further on. Okay, I'm going to just stop here. This all of this is described in these papers, and I want to thank the postdocs and the students who did, especially mostly the biology that I showed you today. And we have some amazing collaborators for all this, and of course our funders over the last 30 years. And just three disclosures, because there are companies that have spun off from some of our work that are funding some of this. So I'll stop here. Thank you so much.